Hello everyone, my name is Leila Oswalt, and today I'll be presenting about my research that I was fortunate enough to conduct this past semester due to the generous funding and support from the Center for Undergraduate Research and Scholarship and Department of Biological Sciences. I'm extremely grateful for this opportunity, and I look forward to continuing my work with CURS and the biology department as I further my research about the use of bacteriophage to treat multi-drug and resistant E. coli infections. I'd first like to briefly discuss my inspiration for this project. Last fall, I learned a lot in my microbiology course about the issue of an upsurge in multi-drug resistant pathogens due to the misuse and overuse of antibiotics. Most people assume that this is the result of doctors prescribing antibiotics too frequently for afflictions that either don't require antibiotics or require a different kind of antibiotic. While this is true, the largest contribution to the upsurge in multi-drug resistance actually stems from agriculture and farming. Due to overcrowding of animals such as chickens, cows, horses, etc., farmers have resorted to incorporating antibiotics into their animal feed to prevent infections from initiating and spreading across barns. This antibiotic diet, seen among the livestock, has allowed for the selection of bacteria with mutations that enable their survival in the presence of antibiotics. These multi-drug resistant bacteria are excreted in animal waste, which can make its way into our communities through runoff, wind transportation, or even through the farmers themselves who care for these animals and therefore come in contact with these bacteria daily. The issue of multi-drug resistance has become so significant that it's projected that our current antibiotics will essentially be obsolete within 10 years. To combat this highly prevalent issue, in recent years we've seen a large spike in interest surrounding phage research, as it's possible to use phage in a clinical setting for infection therapy. Bacteriophage are highly abundant in nature, but are so specific that they typically can only infect a few strains of bacteria. Bacteria can also evolve to be resistant to phage as they can antibiotics, so it's vital that research surrounding phage therapy is conducted. For my project, I decided to look into phage that can treat urinary tract infections, as these are the most common type of infection that relies upon the use of antibiotics. UTIs are most often caused by uropathogenic E. coli, which can make their way into the urinary tract from the epithelia of the gastrointestinal tract. The most common virulence factors of uropathogenic E. coli are toxins, siderophores, and adhesins, which are actually the most important factor in determining their pathogenicity. For this study, the uropathogenic E. coli strain CFT073 was used. This strain holds a genome similar to Shigella, which is a type of pathogenic E. coli that secretes the adhesin called shigatoxin. Water sewage samples were gathered from Windsor, Canada, Perrysburg, Ohio, Archbold, Ohio, and Upper Sandusky, Ohio. These samples were spread plated onto McConkey agar plates, which select for gram-negative bacteria and distinguish lactose fermenters from non-lactose fermenters. Because E. coli is a gram-negative bacteria and most of its strains ferment lactose, well-isolated colonies that had grown on the plates and appeared red in color were touched with a toothpick and streaked for further isolation on both McConkey and hectoenteric plates. On the hectoenteric plates, colonies that appeared orange in color could be further identified as lactose fermenters. From the tertiary streaks of bacteria with different phenotypes such as morphology, mucus versus non-mucus producers, strains that appeared to be Shigella, etc., a well-isolated colony was touched from these tertiary streaks with a toothpick and then streaked on a simple T agar plate against the toxin colicin E1. This toxin is produced by E. coli as a defense mechanism against other E. coli in competition, and therefore would inhibit the growth of any isolated strains from the samples that were genetically similar to or equivalent with E. coli. For this test, laboratory strain TOL-A was used as a negative control, as this strain possesses a mutation that allows it to grow in spite of the presence of colicin E1, while the laboratory adapted strain W3110 was used as a positive control and was expected to be inhibited by the toxin. So here we can see an example of what all the plates actually looked like after spread plating the sewage sample onto the initial McConkey plate. This is one of the plates from one of our first samples gathered from Perrysburg, and again, all the colonies present on this plate are gram-negative, and the red colonies are lactose fermenters, which is what was then used to further isolate new environmental strains. So from all the initial plates, we decided to continue to isolate 6 specific strains that had various morphologies and a good variety of mucus versus non-mucus producers. What was interesting about strain 6 was that while it didn't ferment lactose, as shown by its colorless representation on the McConkey plate and its green representation on the hectoenteric plate, was that it didn't produce any black coloration on the hectoenteric plate. Black coloration would indicate the presence of Salmonella. However, because strain sticks didn't produce this, it could be assumed that it was genetically similar to Shigella, which again is a type of pathogenic E. coli. So we thought this was pretty interesting and decided to keep working with it. But if we look at the regular T plate on the left, this was the test that we ran with the toxin colistin E1. So while it looks like strain 3 inhibited the toxin, we can actually see that right in the middle of the plate where the toxin streak was made, there appears to be a bit of thinning on the growth of strain 3 here. This would indicate that strain 3 most likely produces a lot of lipopolysaccharide capsule material. E. coli are capable of secreting a wide variety of different kinds of capsules as a defense mechanism, and it appears as if strain 3 produces a lot of capsules that got broken down by colistin E1 here. If we look to the left of the toxin streak down the middle of the plate, we can also see, especially from the strain 5 streak, that there's a presence of various spots and plaques within the streak colonies. This indicates that most of the environmental strains were lysogenic and ultimately held viruses within them that became active when the presence of the toxin was added. So this would make perfect sense because most wild E. coli are lysogenic in nature. So once we isolated our set of 6 environmental E. coli strains, we wanted to test all of them, as well as our uropathogenic CFT073 strain, against a series of antibiotics belonging to the class of beta-lactams. Once we combined about 100 microliters of each strain with top agar and plated them onto T plates to ensure lawn growth, we then used ampicillin, piperacillin, and ticarcillin to measure the resistances of each strain by measuring the diameters of the zones of inhibition, or zones of clearing around each tablet. So for each antibiotic that's listed here on the slide with something dash something, this represents a beta-lactam in addition to a beta-lactamase inhibitor. Typically, bacteria are capable of evolving to use the enzyme beta-lactamase as a defense mechanism against beta-lactam antibiotics. Therefore, it's expected that any zones of inhibition surrounding the antibiotic tablets with beta-lactamase inhibitors would be larger in diameter when compared to the antibiotic tablets without their corresponding beta-lactamase inhibitors. So this is just a table comparing all the diameters of the zones of inhibition present on each strain's plate. When completing a Kirby-Bauer test, specific ranges regarding zone diameters are provided for each antibiotic used to determine if the strain would be resistant, susceptible, or somewhere in between to the antibiotic in a clinical setting. A few zones express the presence of halos around them, which would indicate the presence of capsule material, as some bacteria were most likely starting to break down in the presence of the antibiotic before the zones were measured. However, all zones of inhibition were measured as areas of total clearing, not from the halos. Across the board, it appeared that our uropathogenic CFT073 strain was relatively susceptible to all the antibiotics tested, which was anticipated as the strain was a bit older in comparison to the freshly isolated environmental strains. Strain 3 generally represented the most resistance to the provided antibiotic. However, multi-drug resistance cannot be determined just from this one test, as only beta-lactams were used. The strains could potentially be resistant to other classes of antibiotics, but we would simply just need to test this first. Once we had a pretty good grasp on understanding our bacterial isolates, we then began isolating phage from the original watered sewage samples. We first treated all the samples with chloroform to kill any existing cells and then centrifuged them to remove any cellular debris from the solution. From the sample solution, serial dilutions were made by extracting 20 microliters of the original sample and then adding it to 100 microliters of phage buffer. This first solution that we made was our 10 to the negative 1 dilution. From this tube, we took 20 microliters of it and added to it another tube with 180 microliters of phage buffer to make our 10 to the negative 2 dilution. Using our 10 to the negative 2 dilution, we took about 20 microliters from this and added it to another tube with 180 microliters of phage buffer and made our 10 to the negative 3 dilution. And then to make our 10 to the negative 4 dilution, you guessed it, we took another 20 microliters from our 10 to the negative 3 dilution and added it to 180 microliters of phage buffer to make our 10 to the negative 4. So given all our dilutions, we then inoculated about 100 microliters of each with the laboratory-adapted E. coli strain W3110, and we inoculated it with each dilution and then put all the tubes in a 37°C water bath, which is the optimal temperature for phage activity, and left it in the bath for about 10 minutes. Once we took these out of the water bath, and 100 microliters of each were pipetted using the micropipette. We added it with T-top medium and then poured the T-top medium with each dilution onto T plates to ensure lawn growth. So after incubating overnight at 37 degrees Celsius, the plates were observed and well-isolated plaques, typically from the 10 to the negative 4 dilution plate, were picked with a micropipette tip and then placed in phage buffer. The picked plaques were then underwent another round of serial dilutions and were spotted onto plates with lawn growth of W3110 to verify that they were there. So here we can see a few of our plates after inoculating W3110 with the serial dilutions. On the left, we can see a 10 to the negative 2 dilution from an Archibald sample, and then on the right, we can see a 10 to the negative 7 dilution from an Upper Sandusky sample. Most of our samples only underwent dilutions out to 10 to the negative 4, but it appeared that this specific Upper Sandusky sample had such a high concentration of phage that we needed to continue to dilute it out so that the plaques were better isolated for picking. And then these are most of the plates that we used for the dilution schemes of the picked plaques from the original dilution plates. We ended up isolating about 10 new phage that inhibited the growth of W3110. However, we found that the picked plaque labeled EU potentially had more than one type of phage, as it continued to demonstrate a vast multitude of plaques, even out to a dilution of 10 to the negative 6. So once we had a pretty good set of new phage, we wanted to try and identify their potential binding sites on host bacteria. So 2 different laboratory-adapted E. coli strains with outer membrane protein deletions were plated for lawn growth onto T plates. One strain had been genetically modified to have a deletion of the protein binding site called tone B, while the other lacked FHUA. The new phage were spotted onto the plates with the lawn growths of each bacterial strain and then evaluated for plaques. So this table just shows that the phage labeled M1 relies both on the tone B and FHUA binding sites to infect bacteria, as the spots for this phage did not display any presentation of plaques after incubation. Likewise, the phage S1 appeared to really rely on the tone B binding site, while phages UE and ECUS5 appeared to rely on the FHUA binding site, as these spots did not produce any plaques as well. Finally, we wanted to determine each new environmental E. coli strain and our uropathogenic CFTO73 strain susceptibility to both our new set of 10 phage as well as 13 older phage that were isolated by the Larson lab in previous years. So this was done in a similar manner as the previous test for binding sites, as we plated each bacterial strain for lawn growth and then spotted about 5 microliters of each phage onto the plates to examine the presence of plaques after incubation. So similar to the last table, for the previous experiment of determining the binding site, the blue squares here indicate the presence of plaques after incubation, and it appeared that most of the newer phage could not inhibit the growth of the environmental E. coli strains and could potentially also rely on specific capsules for binding sites not secreted by the environmental strains, while being inhibited by other types of capsules that the environmental strains possibly did produce. However, it appears that the pre-existing HR6B phage is capable of inhibiting the growth of all strains tested, including CFTO73. This phage was isolated by the Larson lab about 10 years ago from an agricultural sample of horse feces. Therefore, it would make perfect sense that this phage could inhibit the growth of our uropathogenic strain, as most uropathogenic E. coli and its corresponding phages are not isolated from human sewage, but rather from animal waste. It would thus be most interesting to further analyze HR6B, potentially through DNA sequencing, and ultimately look more toward the realm of agriculture and farming, especially in regard to further isolating phage from multi-drug resistant bacteria. So overall, we found some new environmental bacterial isolates that appeared similar to E. coli. DNA sequencing, of course, would verify their specific genus and species, as well as their specific strain, to determine not only their pathogenicity, but also their resistance to antimicrobial agents, such as antibiotics. We also were able to isolate some new E. coli specific phage. However, it appeared that most were unable to inhibit uropathogenic E. coli. But again, only DNA sequencing and analysis could confirm or deny this claim. Fortunately, we did discover that the HR6B phage was able to inhibit all of the bacterial strains tested, including the uropathogenic CFT073 strain, which was ultimately allowed us to recognize our next steps in the project. So finally, from our findings this semester, we realized that our next steps are to sequence the DNA of our new phage and HR6B to further identify their methods of host infection and inhibition. If we wanted to work more with our environmental strains that we isolated this semester of E. coli, we would want to test them against other classes of antibiotics to determine if they truly are multi-drug resistant. Our next biggest step forward, however, will definitely be working further with HR6B and focusing the project more on bacteria and phage isolated from agricultural samples. Again, I am most grateful for CURS and the Department of Biological Sciences for granting me this opportunity to initiate my own project and continue to learn more about this most prevalent issue within the biological and medical communities. I hope to continue this project in upcoming semesters and add to the plethora of literature surrounding the topic of phage research in regard to uropathogenic E. coli infections. Thank you all.